Hello, everyone. Welcome to this first Signals Notebook Quarterly Connect of 2023. My name is Rochelle Beanstalk. I'm the Product Marketing Manager for the
Revvity.
Signals Notebook, and I'll be your moderator for this first Signals Quarterly Connect session. This ongoing Signals Notebook community series is an opportunity for you to learn about this newly added functionality in Signals Notebook and hear how others are using the Signals Notebook to improve their productivity and data transparency through enhanced collaborations. This particular Signals Quarterly Connect has a topical focus on biologics and the Signals Notebook. This is our first Signals Quarterly Connect for 2023. Our previous sessions have all been recorded and are available for streaming on our website. Future dates for Signals Quarterly Connects for 2023 have been shared along with this registration. So I really hope you'll join us for the future programs, which are scheduled for May, August, and November. We want to continue to keep this series as interactive as possible. So please submit future topic ideas in the chat box, or please reach out to me in between the sessions with your input. You can email me at my email address, which is shown, with suggestions or comments regarding the Signals Quarterly Connect sessions. A couple of quick housekeeping reminders before we start. We will briefly open up to questions after each presentation. We also have ample time at the end for additional questions and open office hours for general questions regarding the Signals Notebook. Please submit your questions through the questions box located on the right-hand side of your screen. Lastly, this webinar is being recorded and the recording will be shared with all attendees in the upcoming days. So now let's jump to our agenda and program for today. So we have two speakers for today. Our first speaker, Signals for Biologics Workflows, will be Lawrence Mulcahy, who is our product manager for biologics in the Signals suite. Our second speaker will be David McClimate, who's head of automation at Horizon Discovery, who will speak on expanding the Horizon services at Horizon Discovery. Now let me introduce Larry, our first speaker. So Larry Mulcahy is the product manager for Biologic Signals Suite. His scientific training and research background includes biochemistry, molecular biology, endocrinology, and academic drug discovery. He spent the last several years in software as a scientific solution consultant across industries with a heavy focus on biopharma R&D. So now, Larry, let me hand it over to you for your presentation.
All right, thanks so much, Rochelle, and thanks everyone for joining. Really excited to share some of the capabilities of Signals Notebook with you today, and hear some of the questions, and maybe gather some feedback from you guys for the future. So, with that said, let's get started here. So I have a really brief agenda here. I just wanted to set the table of the Signals platform and Signals Notebook and how that informatics is ultimately geared at helping you do better science, right? It's really intended to assist you, and just talking about some of the capabilities and how these tools can help you. After that, I'll give you a little introduction to Signals Notebook. I know some of you are current users of Signals Notebook. Some of you are maybe new to the platform or don't have access, you're just curious. So we'll introduce some features and just some general things that you may or may not be aware of, just so everybody's at a same level set. And after I do that, I'll give you a little demo in the platform, just so everyone has a chance to see a few of these capabilities, and we'll go from there. All right. So informatics for better science. I think, as the introduction states, I was in labs before myself, and I used the handy dandy paper notebook that often looked exactly like this clip art you see here, right? It has writing, it has a gel image taped into it. I can't imagine they're still using those thermo printers still, but maybe they are. And for the individual user, this works great. And the reality is, for a lot of what you do, you're still using things like this in the lab because you're not going to have a keyboard or a device potentially in a lot of places in which you work, right? I used to do all kinds of flow cytometry There was a computer in there for that, but heavily restricted use in terms of how it was connected. So the intention is we're providing you a place to capture the experimental narrative, the key data, the things that you're going to come back to. It's almost like building a better notebook by putting the key parts into Signals Notebook. And one of those things that I've often thought about when I'm talking to prospective customers and when we're designing things, it's all around a data model. And I know when I was an academic, I didn't really think about a data model, but even without thinking about it, we're all doing it. And it's often through a tool like Excel. You're keeping track of strains, you're keeping track of plasmids, and Excel and the Office suite is omnipresent. So it's often the easiest tool to use to start making your own database. Something like that doesn't have a lot of business logic. It's not super configurable. It may not be consistent across the organization. So when you're talking about a company that goes beyond a few people, this is often not a scalable solution. You need something better to track your critical things like a plasma database. And coming down to those molecular items like the proteins, the plasmids you're designing, and anything else like that, you want anywhere you're putting these things to have some capabilities to interpret the files that you use to generate this, right? So an Excel plasma database might have a link to the files that are somewhere on a shared drive or something like that, but you can start kind of fragmenting your informatics environment if it's not all into context. And importantly, your solution has to have some sequence capabilities. And then you're doing collaboration, right? Email is often the easiest tool to use in terms of handing off tasks. I've been in some lab environments where that really is the way to hand off between teams. And while email is great, we're probably all getting far too many emails, and it's easy to miss something, right? It's not in the context of your daily workflow. And that, coming back to that gel picture, there is a lot of rich type of data that you'd like to preserve, right? You might do an analysis of a Western blot or a Kamasi stain gel for something you can put into tabular form that would go into Excel, right? But coming back to the original image, making it searchable in the context of an overall experiment, that's a tricky challenge and something that a notebook has to do if you're going to go away from paper to an electronic solution. And then, of course, you need flexibility in your data modeling because you're creating all kinds of things in the biologic space, right? Viral vectors, engineered cell lines. The list of modalities continues to expand, so your platform has to have flexibility so you can capture the data, coming back to that, data model concept of the Excel spreadsheet, the way your organization values it, what's important, what's tractable, keeping the important components all together. So to me, the critical components for this, and this is across the product team at Signals, is we want to make our tools easy to use. I think we can always get better at that, but I think generally most users, when they come into Signals Notebook, find the platform relatively easy to use. I think conceptually, the way it's laid out, the way you add content to it, we'll see it. But that's key because if anything's too complicated to use, you can think about this with any product in your life. You're probably going to start to not use it to its fullest capacity or maybe not even use it at all. So, we definitely don't want that. So we want to make it, highly functional but easy to use. And then search is so key. Coming back to that problem of finding information when someone joins an organization, when somebody leaves an organization, when your organization is large scale and spread across multiple sites, search is so key. And, this is a transformation that continues. It's probably led on the consumer side by companies like Google, right? When I go to my Gmail account, I don't organize my mail anymore. I search for it. There's far too many messages. And I think that's a concept that Signals Notebook has delivered quite well. Sequence intelligence is important as well, coming back to the plasmids, the proteins. I'll show you a little bit of that in the slides and the demo. So we do handle several universal formats for DNA and protein quite well. We're going to expand the modalities that we support in the near term, and that's a major focus actually of this year. And then I think all of this gives you better collaboration, right? Instead of potentially fragmented emails and things of that nature, you can start working in an environment where everybody is. And I know Dave is going to talk a little bit about that in his portion, but I'll share some bits here too in terms of how this can be collaborative. All right, so the brief introduction, for those of you that that don't know Signals Notebook at all, one of the really nice features, like many of the consumer products you use, you just log into it with a modern web browser, right? If your organization has certain ways of logging into common applications, you could do that with Signals Notebook too. It's, wherever you are with an internet connection, you can get to it. And it does integrate with certain desktop applications, some of which we produce. You might be familiar with, like ChemDraw. There's a seamless integration with that. And we also have an integration, which I'll show you, with a molecular biology tool, SnapGene. So there's this concept of, while we're in the cloud, for those applications that are not yet there, we try to partner with and build out, ways to make the experience still fairly seamless. And when you log into Signals Notebook, part of the context here is giving you a dashboard. So it's kind of your personal view of the information world of your organization where you're working. My dashboard is fairly basic here, with like kind of the standard configuration, but it's very customizable. So you can populate different items here. Like if you had work in queue, and you see some of those examples on this screen, like things that are waiting for me to review, things that are in review, recent items that I've worked on, and you can also favorite things. So you can come back to them, be those, notebooks or other kinds of things. And critically, each user can customize their notifications. So coming down to that collaborative element, when someone sends something to you to review or someone tags you in an experiment, you can see that in the application. And I know I maybe denigrated email a little bit, but for those of you that want that too, you can also get notifications via email, right? So there's two ways to make sure you're up to date on information in the system. One of the biggest things that I think is really nice here, and I'm showing an animated picture of this, is dropping content into an experiment. So you think about a lot of experiments that are done in the biologic space. Maybe you're ordering things from vendors, and I have examples of 10 plus files being dropped in. Some of those are Excel, some of those are PDF, and you see for documents we support, we render a preview really quickly, and that includes for the plasmid vectors as I scroll down, right? You see an order form from GeneWiz for maybe the creation of these plasmid vectors. So content is really easy to bring in, and there's lots of types that we natively support, and really, really critical, a lot of that information is indexed directly for search, be that just the document metadata itself, but also the information within the document. So kind of all that complex data you're dropping into an experiment, it can be found, right? So I called this section, if you can see that, QC documents, just kind of those fragments that may get lost. So you could think of this as like storing all of that key information into the experimental context. So if you ever need to come back to it, it's as simple as a search to find it. The other thing that I think helps with adopting an informatics tool like this is templatizing your common workflows. So once a tenant is set up and configured, which can be done by an admin all through the user interface, so no custom coding or anything like that, you can build out templates to capture common workflows. And this one I'm showing here is a snippet of a workflow for ultimately doing in vitro transcription from the design of a DNA template onto the creating the RNA prep. And you're seeing one little snippet of this template here. And that snippet of the template is showing two parts. One, it's a table within Signals Notebook where you can run calculations. So actually, there's a field here where the user doesn't input information. They input the data they have, and then a calculation is done for them. So kind of those repeatable things you're doing, like a DNA concentration, transfection, reagent amounts, those things can be captured easily. And then below this, this example, a customer was asking me if they could put a protocol in from New England Biolabs, and we just copied and pasted the protocol from their website, linked here, right into a text element in Signals Notebook. A lot of the formatting comes over quite easily. So your protocols, your SOPs, tables, all of that stuff can come in here. And you saw on the drag and drop, all of your key content can come in here too. I mentioned search, right? So Signals Notebook is built on modern search architecture, and that's really critical because we want it to be fast, we want it to be performant, we don't want you coming to the notebook, running a query, and letting it hang, right? That's a frustrating experience, and I think those of you that have used our search know that it is fast, right? And the example I'm showing here in this animated picture, is essentially running a search for a feature of this plasmid vector, well cut, an uncommon word, right? And you can see just with a simple text search, I'm able to find that feature that's part of the file. You'll also notice this sequence intelligence component, right? As I go into the hit that I see that well cut sequence, I go into it. I have a dynamic view of the plasmid with the sequence, the map in a side viewer, and all of the features displayed, right? So this is what I was saying about, it changes the way you think, right? Put all your information into the experiment and don't think about tagging it, organizing it in folders and things like that. We have a notebook structure and we have ways to organize, but generally the way Signals Notebook is architected, you need to do a lot less of that pre-planning, and there's a lot less point and click exploration to find your files, right? It's not an old school kind of file structure. All right. So with that said, let's flip over from the slides actually over to my instance of Signals Notebook. And that's the dashboard that you saw on the slides here, right? So just to show you, you could customize this. There's the configuration here. So if you wanted something a little more than what I have on my basic tenant here, you can configure this. The organization of the dashboard just lets you return to the things that you need to frequently, or coming back to this concept of notifications in the platform, seeing anything in here like someone sent me an item to witness, right? We saw that. These things appear in here and can be favorited in the in-app notifications. And I'm going to return to a experiment here that I showed a few snippets from in the slides and just walk you through different types of content here. And I use that word purposely because you're generating all kinds of data, and really that just comes down to different content types ultimately. Images, table, data in tabular form, the plasmid files, all of that. And you'll notice I did a drag and drop in the slide presentation, but the content types we natively support and things that we do with the platform are all present from this content menu. So you can add any type of content, and much of it is natively supported, right? The Office suite is natively supported, so a lot of the things you're used to using just work really well inside Signals Notebook. Now for me, this experiment is starting off with one of my pages here, which is my run planning and results. And you see I have general protocols and just some sequence examples in case people had some questions about that. This is a task, actually. So this is, I mentioned email as a way to collaborate. A task in our system is a way to kind of formalize handoffs between people. And it can be configured and highly structured to include all the information you need. Now, Signals Notebook contains features like inventory, so that if I need to know the specific, down to the container of a material I'm supposed to use for running this protein expression, I have that all within the context, and I'm able to get to that and run kind of standard inventory type of actions like reserving a container, checking it out, decrementing amounts, and the like. Did the back button there. So this is one specific type of organizing workflows in the platform. Coming down to sequence intelligence, here's a Snapgene file that I placed into my notebook. You saw me search for that item, right, when I opened up the search bar. And when you go into a full-size view of this, you have the plasmid map as well as a complete view of all the sequences. So you can see come in here and do things like copy sequences if you need to, copy the amino acid sequences, grab a GenBank selection. There's a lot of functionality within this tool. And importantly, I called out those connections to desktop applications. We do have an integration with Snapgene. So if you do have a license of Snapgene, you can open up your desktop client, if you have it where you're working, edit this sequence, and return it. If you don't have Snapgene, there's still really easy ways to work with these files inside the notebook. You can actually just use this replace. So if you have a new version of the file, you're not using an application we support, you can just use this option to update the file, and all of that is saved in the history of the item itself. So I think that's another important thing here. When you're creating items, when you're updating them, when you're editing them, we have all of those versions saved here in the platform that's done automatically for you. So there's no risk of losing data once it's in the system, right? It's a very robust architecture. So that's another key element I didn't call out, but an important one. As we go down, there's some basic things here like text elements I called out. I have an example of a transfection calculator here, right? This is the table example I gave you. We could put in another flask if we want to. We'll call this flask A and just show you these can be configured to run kind of those standard calculations you want for finding how much lipofectamine, how much DNA do I need. For those of you running plate-based assays, we have a plate element, so you can, map your plates, specify what's going into them, concentrations, replicates, all of that good information. And these things can be copied out, they can be printed if you needed to take them to the lab bench, and they can be sent to other parts of the Signals platform as well for maybe analysis and things like that. As we come further down in the experiment, here's an example of an image, but I'm going to show you another example of an image here with some features that I think are really nice, particularly for these type of documents. So images are natively supported in here. There's a number of file formats we take, but one of the things you may notice is that this image is annotated with kind of common labels, right? The supernatant, the flow through, my fractions, and I've actually circled those. And one of the things you'll notice as I click on any of these, you see in this tabular form here, this is highlighted. So this is fully searchable. This type of content on an image that you annotate within an experiment can be searched directly by you. So again, if you're using common things that come up in every image, well, you'll find a lot of them, but you could filter it down by creator, by date it was created, and quickly find that image, right? So these things that could be floating out there and really hard to come back to, when you annotate it in here, you're getting all of those advantages of our platform. And just to walk to a couple of other elements, because this is an experiment, right? And I talked about building your database and tracking your things. So I've shown you a plasmid in a database, but you see one of these references in my task coming back to the top. It has a unique identifier on it. And if I were to actually navigate to this, what you'll see, it's a subtle thing, but I've transitioned to a different part of the platform called materials. This is essentially the registration. It's the database. And this database lets me track lots of my expression vector here down to the container, to the inventory level like you saw here. And importantly, I can track all of my plasmids in my library. So just like you saw in the experiment, this is a registered expression vector. So it has all of those characteristics, and you can set it up so that two people don't register the same vector if you're making it again, right? Like, has somebody already made this thing? Those types of things are fully tractable. And this is just a point to show you that the sequence intelligence goes across the experimental down to the central registration system of your platform. All right, so that's a really quick whirlwind tour. And let me just wrap up here. Hopefully I've given you, just a taste of some of those features that are for the biologics workflows. And you've seen some of these elements here of the ease of use, the sequence intelligence, the collaborative element with the notifications, the tasks, things like that, and the searchability. So thanks for watching this section, and I'm happy, of course, to take questions as they come in. So let me check out the question panel here.
That was really fantastic. Thank you so much, Larry, for that great presentation and in introduction to the use of the Signals Notebook in the biologics environment.
All right, so I do see a couple of questions here. So the first one I see is, can more than one person work within an experiment? The answer is yes. There is sort of kind of a standard thing with authorship and how your organization handles authorship, who should take full responsibility for an experiment. But we have some signing workflows if you're working in that type of environment. And as long as someone has access privileges, you can have multiple people in an experiment, no problem. Second question I see here is, can you search by the underlying sequence? So the answer to that is we're working on that right now. That's actually a feature that's in development. So you probably noticed the search example I showed was searching a property of that plasmid. I searched the well cut feature. So all of those features are basically text that we well index right now, but we are working on building out so you can search for, an exact match of a sequence. You can run a blast search, a nucleotide or a protein type search. So while we don't have that yet, it is something we're working on. And I think that'll be really critical as another way to find all the experiments where a key sequence has been used, all the, database items to find something similar if you're working on something new. Um, and then I see a question here. Can a user edit the sequences in an experiment? Um, that's a good question. So the answer to that is yes, but I would say with a caveat. So I showed you the integration with Snapgene. So in a way, if you're hooked up to an editor like that, you can open up the application, make changes to it, and synchronize that back to Signals Notebook in the cloud. No problem. You can also upload and replace a file, but our tool is not a sequence editor itself. So it's really a read-only tool. So our philosophy so far is design your sequences with whatever tool you use, whatever pipelines you use, and then bring those into the registration system or the experiments. Um, let's see. And then I see another question here. Are there limits to the data we can add to the signals experiments? How to add RNA-seq data? Is there a link to the cloud, raw data to put in? Yeah, so there are some limits on file size of what you can load in. And what I would generally say for high content data, high volume data like RNA-seq or high content imaging data, it might be best to place a link to that. That's something we could discuss further. The notebook does handle pretty large files, and the tenant can handle lots of data. It's just a question of, is that the right place? And I think you hit that on the head there. So I don't remember those file limits offhand. I know they're in the user guide, but we could probably follow up with that if you can't follow that in the user guide. I see another question. Is there OCR function that is allowing search of the documents? Good question. No. So if your document isn't in a format where the characters could be scanned, which a lot of PDF, Word documents, that's not a problem. But if you were to put it in an image file that has text, we are not OCRing that type of information and making it searchable. Uh, let's see, is there an in-table searching feature? So when you're filling a table on the template, it helps you. Table on the template, it helps you. Oh, I think I follow this question a little bit, and the answer to that question is, in a way, yes. There is a bit of that. Um, Depending on the configuration, depending on the data you wanted to fill in, we have things like hierarchical attributes. So you can imagine if you had like a project and sub-project codes underneath it, and you were filling in a table, and let's say you pick project A, the next column of the table would only offer up options that go under project A. So that's one example, but there's more like that. So if there's any need for, follow up, certainly let us know. Someone's asking, could you please demonstrate adding a reagent into inventory and how it can be used during an experiment? Uh, that is a great question, and I think in the context of time for this session, Um, I don't think it would be good for me to go into that entire flow, but definitely reach out to us, and we'd be more than happy to have whether it's me or one of my colleagues show you that kind of capability with our inventory, because I realize I did a really brief demo of that. Let's see. Is the material inventory capability part of the main Signals ELN module or a separate module? Inventory is part of Signals Notebook. So yes, it ships with every tenant. Your admin has to activate it and give you privileges to access it. So depending on your organization's policies, maybe you're using another inventory application. I would just encourage you to contact your admin to see if you would like to add that. See a question, can you generate a report? Um, I think yes. So you can print an experiment and all of the attachments with Adobe Acrobat can come out. So in a way, that's a report, but I have a feeling you're meaning something more like a PowerPoint presentation. And it's something we've thought about, but there isn't like a push a button and make a PowerPoint feature as of yet. We do let you, come into the platform, find the things you need, download them. So there's some steps to do it, but it's not a push button option. I see a question. Can you annotate protein sequences with the same level of detail as plasmid sequences? Um, so I should be clear. In our platform, you don't annotate these sequences, but with your tool, you can annotate proteins with as many features as you'd like. Generally, there's less automatic features like you saw in our tool, like the restriction enzyme sites and things like that for proteins. But whatever annotations you do in a tool like Snapgene, you could do on a protein as well. Uh, uh, let's see. There's, I see a bunch more questions, and Rochelle, you can let me know when I should pause for the next part. So I'll do this one. Uh,
Why don't
Quick
we just
question.
take one more, and then if we have time at the end, at the very end, if there's some time, maybe we can come back and answer some of the other questions.
Okay, sounds great. So I'll do this last one. Quick question. What level of audit history have we got on the ELN, especially values changed in the tables? Yeah, great question. So the audit log access is an admin level feature. So like all the changes that are happening in Signals Notebook. And the answer to your question is every change like that is in the audit trail. So I showed you the history of the plasmid, for example. So at the end user level, for every piece of content you put into the notebook, you can see all the changes, you can revert them. And for an admin across the whole tenant, they can search and find all of these events. But it is a great question, and the audit trail is fairly granular. So generally, the answer to your question is yes, you can find those table level changes done in the notebook. All right, so I'll pause there, Rochelle, and we can continue.
Thanks again, Larry, for the great presentation and answering all those challenging questions there. So our second speaker today, let's see, is David McClimate, and his presentation is expanding the horizon services at Horizon Discovery. David is the head of automation at Horizon Discovery, and he leads the platform innovation team, which brings together cross-functional skills in automation and assay development and bioinformatics and LIMS and software development. He integrates the disciplines, meeting untapped opportunities and capacity in biotechnology applications. He previously designed and built a metabolic pathway engineering system for yeast and E. coli at the London DNA Foundry and co-located at Imperial College London. The AMOS system allowed the design and automation and build and test of functional metabolic pathways to produce high-value chemicals. Before that, he was head engineer at the Chemical Biology Platform in Oslo, which is the National Center for Chemical Biology with a focus on immunology and personalized medicine. And he worked with automated electrophysiology at Zinchen Discovery in Cambridge, UK. He holds a PhD in molecular pharmacology from the University of Nottingham. And thanks so much for joining us today, David.
Hi, my name's David McClyman, and thanks for the opportunity to give this presentation to you today. So the title of my talk is Expanding the Horizons, Services at Horizon Discovery, where I'll go through some of the offerings that we have at Horizon Discovery in the services unit, and also discuss some of the challenges and solutions that we came up with along the way as we opened up a second site to offer standardized services. Services at Horizon Discovery can support you at any stage of the drug discovery process. From basic research all the way through to clinical trials, our services are built upon our long history in gene editing and cell biology and fall into these four main pillars. Functional genomics uses gene editing technology to understand and interrogate the biology of novel targets across pools or in arrayed plate-based formats. Immunology involves screens that determine the effects of therapeutics on the human immune system. Cell panel screening uses large numbers of cell lines where we assess the biological impact of oncology therapeutic candidates, and that can be done in single agents or in combinations. And finally, we also manufacture gene-edited cell lines, which act as a robust model for reliable and predictable results, no matter what your research is. So I'll just go into a little bit more detail about some of the approaches we take to offering services at Horizon. For cell panel screening, we have an extensive library of over 1,200 cancer cell lines, but for our immunology platform, we can also use primary immune cells from multiple donors. And when using primary immune cells, having robust quality control procedures is incredibly important to ensure cell morphology and cell markers are maintained in vitro assays. We focus on developing automated, semi-automated, and miniaturized pipelines to have experimental consistency and also have high throughput. We have a number of different high throughput readouts such as flow cytometry, HTRF, and NGS, and this produces a lot of data. And in order to produce robust data sets, we use custom bioinformatic pipelines to produce data for things such as target ID, generalized screening assays, and any custom projects that you may want to discuss with us. So just a little bit more about me. I'm Head of Automation at Horizon Discovery, and that responsibility covers lab automation, high throughput work cells, and the assay development to get those processes working well. But what I think is really important is to bring into that group the configuration of SAS, ELN, LIMS, and lab execution systems, as well as the exploration of data analysis pipelines, because by doing so, we can create really advanced screening and manufacturing platforms. And over the 10 years of my career, ultimately, I think what I've realized is that, developing robust scientific systems like this ultimately involves moving tiny little bits of liquid from one place to another. But where I think there is a massive opportunity to unlock scale is to think about what liquids need to move where and when, and abstract those processes within a data model that you build in software. And that's why I call the team Platform Innovation, because we try and bring together a range of skills, working with cross-functional project teams in order that what we're building is the best use of technology and things aren't invented and thought about in silos. So how can we ensure that we build the best systems possible? I think growth is often limited by the development and use of legacy systems developed often organically by scientists to meet their immediate needs. People naturally build on what they have available, which is often just Excel spreadsheets, or if people have the skills, they can make their own custom software. On the flip side of that, lab automation is effectively a solved problem. The technology that we have available can run thousands and thousands of experiments easily, but I don't think it can be effectively utilized without better underlying informatics. So the use of the Excel spreadsheets and the custom software leads to an accumulation of technical debt that can't keep up with the automation, and unfortunately that also embeds behaviors and methodologies that make change difficult. That is why it's important to think about an informatics strategy early on that's appropriate to the problem at hand. For our system design approach, the first key thing to do would be to design a data model that captures the scientific domain effectively, not only in what you're trying to deliver now, but also in a flexible way to add new capabilities and requirements as they come up. With that data model in hand, automated and semi-automated systems can capture and upload that data and metadata directly into LIMS and ELN systems. This is now a more easily realized possibility, as native software as a service options make this approach easier and faster, where the sample tracking, data analysis, and visualization software is accessed through the cloud and the data is connected to those systems directly. Defining that data model can be hard, but it's key. But if this approach isn't possible, then at least having an informatics plan like the basic use of ELN at the very beginning can help. as it's a problem that will need to be dealt with eventually, and the later is left, the harder it will be to undo all those choices. So in order to ensure that we get the best data model possible, we use the platform innovation approach, as that tries to solve problems in multiple domains simultaneously, not in isolation, because I think that leads to more innovative and efficient solutions that are step changes above current states. So that involves improved lab methods such as assay development and process improvement, getting consistent scalable results in microplates, together with the appropriate use of automation from scientists interacting with standalone liquid handlers up to fully automated work cells, and together developing LIMS and sample tracking and ELN solutions closely with the automation is a powerful combination. And when things can't be bought, sometimes you have to build your own software to give you a competitive advantage. And our team tries to translate the scientific requirements into buildable software components, user stories, and features by understanding and prioritizing what needs to be done to ensure the best use of resources. And when all those things come together, that's when things can really take off with data science and AI, because we've got good quality accessible data generated by automation work cells, which allows us to make decisions at speed with the results that we have. And so on to the problem at hand. And what we wanted to do was we wanted to set up a new physical site to help us deliver standardized services. And some of the things that were important to us were to make sure that we had secure project management, firewalling that information off against other areas of the business. We wanted to have good, secure data transfer protocols. In particular, flow cytometry files can be quite large, and HTRF data processing can be quite complex. We wanted to make sure that we had good collaboration between those teams and effectively manage the team and the project in an efficient way. And we also needed a system that could interact with our existing cloud-based analysis tools. So it was obvious that we needed to have some sort of cloud-based software, probably an ELN solution to deliver this. Horizon can perform lead optimization in two modes of immune cell assays. These can be purified primary cells or a co-culture of two types of cells. The monoculture approach helps us understand the sole effect of a drug on a specific primary immune cell type, whereas the co-culture approach can explain how the compound affects the immune microenvironment. The standard mixed lymphocyte reaction (MLR) assay is a co-culture one-way T-cell proliferation assay. T cells from one donor will proliferate in the presence of antigen-presenting cells, in this case monocyte-derived dendritic cells from multiple donors. Then the co-culture of these causes a histocompatibility mismatch between unrelated donors, which provokes an immune response from the T cells. Checkpoints are engaged when a T cell binds to an antigen-presenting cell, such as a dendritic cell, and immunotherapies, also known as checkpoint inhibitors, target this synapse between T cells and antigen-presenting cells, specifically to block the immune checkpoints that usually switch off T cells. Measuring MLR in the presence of an active compound is a useful method for determining the immunomodulatory potential of that compound. We at Horizon, we offer a multiplex readout, so where we offer both data on the proliferation and activation of T cell subsets measured through flow cytometry, and we also provide data on cytokine release assessed through HTRF. For those that don't know how specifically these assays work, I just wanted to go into a little bit more detail on the different readouts we use. On the left-hand side is an example of flow cytometry or FACS data. Cells flow through a series of lasers one at a time, and different measurements are made depending on how that light interacts with those cells. Fluorescent antibodies can be used to mark cells that have specific surface markers, in this case CD25. On the left-hand side, the x-axis represents the amount of fluorescence associated with CD25, and the box represents the regions that would have points in them if the cells express that surface marker. Where the left figure there has no cells in the box, the right figure shows a large population within that region, showing that compared with controls, there are now a large number of cells expressing CD25. On the right is a graphic showing HTRF, time-resolved fluorescence, which allows us to measure the presence of certain molecules within an experiment, in this case cytokines. When a donor and acceptor molecule come together, they release light that can be measured in a time-resolved manner, measured in plate readers such as the Envision. The standard MLR assay is a flexible assay for early drug discovery. It can be useful for drug developers who must evaluate the safety of their compounds as part of the clearance process through the FDA or the EMA, who may request validation of in vitro safety assays before moving to clinical trials. And the MLR is a fast and efficient way to move towards those trials. For drug developers in oncology, it's important to determine the effectiveness of T lymphocyte stimulation in the immune microenvironment. The MLR assay can determine the activation of lymphocytes in an allergenic reaction, comparing it with the gold standard drugs in the industry. Finally, the MLR assay has also been optimized to evaluate the inhibition of immune activity, so drug developers in the areas of autoimmunity, transplantation, and inflammation can compare the effects of lymphocyte inhibition compared to standard inhibitory drugs. So just to recap, what we wanted to do was to use the Signals notebook to help us transfer the execution of our standard mixed lymphocyte reactions to a new site that we were opening up. And what we wanted to do is to make sure we had the right informatics infrastructure from the very start to ensure this project was a success. We required departmental and team organization to have granular permissions for experimental notebooks, even among the immunology team. So if you look at this figure here, within Horizon Discovery, like many companies, we have multiple departments, one of which is services. Within services, we've got multiple types of services that we offer, of which screening is one. And within screening, we have multiple groups, of which immunology at one. And at that point of the tree, we even needed to have granularity between the existing immunology team and that at the new site. The metadata fields on a given notebook are used to assign certain notebooks to certain teams and departments. These assignments are used in combination with the system security policies in order to ensure that only certain notebooks can be seen by certain users, depending on which teams they are assigned to. As setting up a second site was new for Horizon Services, we needed to find ways to enable close collaboration and management cross-site, which is something that was new to us. Even though due to the COVID-19 pandemic, we're all more used to working remotely, we wanted to explore what tools we could use to make that collaboration in the lab work more smoothly. The comments pane within experiments is really useful to enable asynchronous chats with the scientists within the context of that experiment. Having the conversation there is better than using, say, Microsoft Teams or email, because you're already in the context of that experiment. So a lot of that context that we need to provide, if you are using something like Teams, you don't have to, because you're just talking directly within the page. A second feature that's a little bit more formal is the use of tasks. You can assign specific tasks to specific scientists to do and put a deadline on those tasks to be completed. These act as a more structured way, I think, to deal with things that need to be done in the lab and are a little bit more formalized than using the comments function. The task has a lot more granularity in the sense that they have to be actively marked as complete when they're done, which helps enable the management of people and projects within the notebook. The combination of both of these asynchronous management capabilities within the platform, one soft and one more formalized, greatly enhance the collaboration efforts within the teams using it. The dashboard function has been incredibly useful, allowing our team managers to be able to manage their own work, manage the work of their team at the existing site, but also keeping separate the management of the new team at the second site as well. By being able to customize the dashboard searches in any way required, the things that are important to those managers were easy to draw out. Having areas that show the notebooks of specific subteams facilitated team management in a big way. Also, having a view of notebooks that need to be checked and reviewed helped project management, making it easier to drill in and ensure projects were on track and on time. We also placed the SOPs within that platform, making them easily accessible for when they were required in the lab. This acted like a knowledge base so that any questions or queries could be answered easily by having the scientists search for what they need. This was incredibly important when the management of that team were not on site to be able to tap on the shoulder and ask a question to. The SOPs were then used to create templates where all stages of the process were laid out, leaving clearly defined sections where files needed to be placed. We highly recommend the use of templates in standardized environments because it makes it a lot easier to train scientists and ensure everything is added and up to date. Having the files in a standardized place within the notebook also allowed us to connect other systems to it to pull those files for later analysis. As we said, it was really important that any solution we use could connect directly to our existing cloud-based bioinformatics analysis systems. The ISHTAR system that we have can be used for applications such as Sanger sequencing indel detection, NGS indel detection, pooled screening analysis, and innate screening analysis, amongst other capabilities. So we needed this system to be able to connect to the Signals notebook and automatically find files within that environment so they could be analyzed and sent back in the correct format to deliver to the customer. The ISHTAR system uses the Signals Notebook API, watching the notebook for any files that are uploaded by the scientists into the specific sections of the template. That is then a trigger to pull those files and carry out the analysis. When it's completed, the analysis is returned to the correct notebook. No manual manipulation or moving of files are necessary, and results are returned in the correct format for the customer. One major limitation of this approach is there's no interactive QC or data exploration possible, and if any errors in that analysis, they have to be sent again to be analyzed. For each project, the analysis produces HTRF and FACS outputs. This includes the results file, annotated readouts, and plots for each donor for each compound. The top figure shows the curves of CD4 positive T cell proliferation in the presence of daglizumab. This plot highlights the consistency between donors and the robustness of our platform when handling primary cells. We also produce quality control charts for each plate, which you can see on the right, to ensure consistency through multiple plates of proliferation and activation. On average, this platform produces around 1,000 figures per project, which are collected together in a zip format and returned to the notebook to be sent to the customer. As mentioned, the bioinformatics tools don't have interactive QC and reanalysis, despite being powerful at what they do. This is why we're really excited about the plates feature within the notebook, as those plate maps and data files can now be pulled directly into VitroVivo. The way this operates is really nice because the spotfires within that Signals notebook know which notebook they were taken from, so they can pull files directly and plates directly within that context of the notebook without having to search through the whole system. Having this direct connection opens up a lot of opportunities to enable us to easily introduce interactive QC and reanalysis, replotting, testing hypotheses without having to maintain our own custom code. So that's why we're really excited about all the latest features in VitroVivo and Signals Notebook in combination and how we can start to utilize them within our analysis workflows in future. And so in summary, the Signals Notebook was essential for our success as we started to deliver cross-site standardized services. The granular permissions allowed us to firewall data between different teams. The data transfer function allowed us to place files within the notebook and have that be shared across sites. And the comments features and the task management greatly enabled collaboration and team project management. The APIs allowed integrations to be made with our existing cloud analysis tools, and we're really excited about the future with direct connections between the Signals Notebook and VitroVivo, which will have a massive impact on our future plans. Being able to bring interactivity, QC, and reanalysis without having to maintain our custom code will ensure that the future projects we add to this platform with new standard assays will be even more successful.
Well, that was really great, David. Thank you very much for that description of an application of the Signals Notebook in the biologics environment. And now we're open to some questions.
Okay, thanks for that. I'll just look through some of the questions that we've got in. Thanks for sending those through. So got one here. What was the best collaboration feature and why? So that was quite a surprise for me because actually, talking a lot about data models and all the different features that that we use, but actually, what the folks in the lab really loved the best was the chat functionality within the Signals notebooks. Being able to kind of focus in that conversation directly onto the actual experiment itself rather than in Teams or email, as Larry said, was actually surprisingly useful just for day-to-day collaboration and chatting. So sometimes even the basic things rather than the most complex features are the ones that people actually use and get the most value out of. Okay, how did you set up the use of the API in the notebook? So like a lot of SaaS native software, the admins can set up an API key. So once you have that API key, set up within the platform, you can basically use that as a connector in whatever programming language you use, either Python or C#. And I think it's really important. I think the use of APIs and evaluating software based on how flexible they are, because really any functionality that you might need that the designers of that software haven't thought of, you have the opportunity to build because you've got that freedom to operate within that space using the APIs in any way you like. And the API for Signals Notebook is fairly fully featured. So maybe just one more in the interest of time. How does the Signals suite extend into the biologics capabilities into automation use cases where requirements for content capture are well-defined, restricted? So that's one of the things that we've, been experimenting with. We've not done a lot of in-depth work in this area, but for instance, again, by having a fully featured API, we can put metadata within structured tables within Signals Notebook, pull that metadata directly into Liquid Handlers and display that information to the user. And then using the API, we can automatically upload Liquid Handler output files back into that experiment. So we think that, that's a nice use case for the combination of, structured information within those notebooks that have to be directed towards Liquid Handlers. And again, it's just one of those things that with a fully featured API, really the sky's the limit on the sorts of things that you might want to achieve. So I'll call it a day there, Rochelle, unless you've got any other questions.
Okay. Well, thank you very much, David, for answering all those questions. And I think we're just about at time. But I wanted to thank everyone, and we have a demo, a really nice demo online that everyone who attends today will receive a link to, which is a biological demo of the Signals notebooks and the biologics environment. And so that you can try out some of those features, and it's a guided demo. So I recommend everyone who has interest to try that demo out. I want to thank both of our speakers today, both Larry and David. Our next Signals Quarterly Connect is scheduled for May 24th, and I hope you'll join us at that time. You can stay up to date on the Signals Notebook new features and functionality via the Signals Notebook What's New Informatics section of the website. And this session, as well as all the previous sessions, have been recorded and are available for streaming on a website at the Signals Notebook Quarterly Connect plus
Revvity Signals.
So that ends our Signals Notebook Quarterly Connect session for today, and I hope you'll join us again on May 24th.